Okay, so I'm going to present what my lab has been working for the last several years. This is to understand how the brain reacts in conditions of injuries and neurodegenerative disorders, and how we could protect the brain from the debilitating effects of these illnesses. And as an introduction, since the audience is going to be undergraduates and graduate students, just sort of introduce you to the different kinds of cells in the brain, which you probably would have heard of. The major cell type are the neurons. So these are the ones that are involved in information processing, communicating between each other and giving rise to both actions at the physical levels and also at the more cognitive level to the extent of learning and memory and even cognition. The next type of cells are the astrocytes. These are the major ones involved in a variety of functions, and this would be the major players of the stock. So I'll come back to them in just a second. And you have the oligodendrocytes. Their job, as you can see, is basically to wrap around the axon terminals and to protect the axons and improve the firing rate of these axons. So improve the action potentials, which normally jump from each of these gaps that you see called the nodes of Ranvier. That's how the action potential jumps when it starts at the cell soma. And finally, the cells called the microglia. These are not in the strictest sense the part of the brain, meaning they don't derive from the same precursor cells that gives rise to the neurons, astrocytes, and the oligodendrocytes. The microglial origin is in the yolk sac. And very early in development in mouse that people knew, and same for humans as well. They actually form in the yolk sac, migrate through the circulatory system, enter into the brain, and take residence in the brain. And these act as the resident macrophages of the brain. So the microglia are the immune cells of the brain. They come from somewhere outside compared to the other cell types that are formed within the brain from the nervous tissue that's developing. But like I said, among the most interesting of all of these cells are the astrocytes. Because if you look at all the other cells, they have maybe one or two critical functions, and probably the only functions that they do, the neurons to fire action potential, process information, oligodendrocytes to myelinate the axon, microglia to sort of have immune surveillance function. But if you take the astrocytes, and these have at least a dozen or more functions attributed to them. One, of course, is they help in neural transmission. They secrete their own transmitters called the glial transmitters. You would have heard of neurotransmitters, but there are molecules such as adenosine and ATP and D-serine, and which actually are secreted by the astrocytes, and they can modulate the function of the neurons. And they actually are in contact with the capillaries, blood capillaries, together with the endothelial cells of the capillaries. They form what's called as the blood-brain barrier, a very protective barrier that prevents anything from entering into the brain beyond a particular size of the molecule. And that poses a problem because most of the drugs you try to target for any illnesses in the brain, it's very difficult to get it across the blood-brain barrier. So, and astrocytes help in maintaining the blood-brain barrier permeability. And of course, they are in constant touch with all the neurons and also oligodendrocytes. And the first term, and these are together called the glia, the oligodendrocytes and the astrocytes are together called as the glia. And the concept was initially introduced by Rudolf Virchow in 1858. And this basically means a glue. And because in the initial techniques that were used to stain these cells in the brain, they looked like cells that were sort of holding the brain together, and hence they were called as a glia. And glia are termed as either macroglia or the microglia. The microglia are the ones that we saw just a while ago. The glial cells, if we take the macroglia, the major ones are the astrocytes and the oligodendrocytes in the brain and in the spinal cord. The Schwann cells perform the same
Hello,
function.
sir.
Yeah.
Sorry to interrupt. Your slides are not being changing, sir. It is not being changed. Yeah, now it is
Okay.
changed to
Okay.
fourth slide. Now it has gone to fourth slide.
Okay. I'm so sorry. So,
Thank you, sir.
okay, fine. So, but is it still in the not sharing mode, right? You don't see it in the full screen mode, do you?
Now it is second slide, sir. You can go ahead, sir.
Okay. So these are the major cell types. I'll just recap quickly. You have the neurons, the oligodendrocytes, and the astrocytes are the major cells of the brain, and the microglia are the ones that actually take residence into the brain very early in development. And if you look at the functions, and the astrocytes are in constant contact with both the neurons and the other cells on one hand, and in contact with the blood vessels on the other to form this tight barrier called the blood-brain barrier. The major idea of what these glia came about from very early work by stalwarts in neuroanatomy, one of whom is Rudolf Virchow, that coined the term glia. And we normally call these glial cells, the astrocytes, the oligodendrocytes, and the Schwann cells are the macroglia, and the microglia are the ones that are derived from the outside. The very first images of these glia came from the staining and characterization by Heinrich Müller. The first evidence of how these cells look came about from this gentleman, and his name is immortalized in the field by giving his name to a class of glia present in the retina called the Müller glia. And if you were to stain these cells in a dish, in culture, you actually see they are stained for an intermediary filament protein, a cytoskeletal protein called GFAP, and DAPI stains the nucleus. You can actually see that these cells have more stellate morphology, more like star-like. And hence, these were called the astrocytes because they have star-like appearance, and that name was given by Michael von Lenosek. And these two gentlemen, Santiago Ramon y Cajal and Camilo Golgi, are the ones that actually revolutionized the way you can actually look at these cells in the brain. By Golgi was the one that came up with this technique for labeling cells using silver nitrate or chromium, potassium dichromate. And you actually can label these cells. And Ramon y Cajal painstakingly drew these cell types from every part of the brain, including the spinal cord, to give us the idea that the neurons are actually connected to each other through these junctions called as synapses, and there are many, many varieties of neurons and astrocytes. The oligodendrocytes are the other cells that was originally identified by Pio del Rio Ortega, who also discovered and named for microglia as well. So, and if you look at the astrocytes in general, we put them into two larger classifications, and one are the protoplasmic astrocytes. These are present in the gray matter of the brain. The gray matter of the brain are the regions where the cell bodies of the neurons are situated. As you know, the neurons sit in one place, send their long process called the axons to wherever they have to project. And the regions where only the axons are going, they are sort of bundled up and they move. And those regions are called the white matter because when you stain them, they actually stain very differently. The gray matter, because of the presence of these cell bodies, actually stain darker compared to the white matter because the white matter is mostly axons and myelin, so it's a fatty sheath of myelin that doesn't stain anything, so it's called the white matter. It's basically the color difference is what the terminology means. If you look at them, they look very different. If you look at the protoplasmic astrocytes, they are smaller, stouter cells with shorter process, but contain a lot of cytoplasm, whereas the fibrous astrocytes in the white matter are thin, much longer. Their process can go for about 150, 200 microns, whereas the length of these processes would be around 50 microns. And also by presence of where they are, the protoplasmic astrocytes are in constant contact with both the cell soma of the neuron and also the synapses, whereas the fibrous astrocytes are mostly in contact with the nodes of Ranvier and the blood vessels. If you look at these astrocytes as it is stained in these tissues or cells, you won't be able to appreciate the complexity of their morphology. How these cells, do they look very simple or are they a little bit more complex? And that came from a dye filling experiment where you actually take an astrocyte, identify an astrocyte in a tissue, and you fill it up with a dye, a fluorescent dye. And these are live cells, so you fill the cells and the entire cell within minutes will light up, and this is what will give you the complexity of the astrocytes are. And if you label neighboring astrocyte, if you label one astrocyte with a green dye and the neighboring astrocytes with a red dye, and they looked at regions where these astrocytes overlap, as shown in yellow here, people were able to appreciate that the overlap is not more than 5 to 10%, indicating that despite having a very complex morphology of branches and finer protrusions and branches, these astrocytes are very territorial, that they do not overlap into each other's territory and maintain their domain very, very strictly. And if you were to compare between a mouse and a human astrocyte, you can see this is sort of drawn to scale to a good extent. A mouse astrocyte is much smaller compared to the human astrocytes. And the difference in size comes about looking at the cell soma versus the branches of these astrocytes. If you look at the cell soma increase, it's just about 1.15 fold. So 1.5 fold is what the increase in the cell soma. But if you compute the area occupied by these astrocytes, the human astrocytes is nearly 3-fold larger in terms of the area covered because of the virtue of the branches that it has compared to the mouse astrocyte. And the mouse astrocyte has been calculated, this is in the cortex, contacts about 100,000 synapses, whereas a human astrocyte, a single human astrocyte, makes contacts with 2 million synapses and about a few thousand neurons. And whereas the mouse astrocytes are in contact with a few hundred neurons. So this is the difference. And the astonishing thing is when you look at evolution and how these glia number changes actually from lower organisms to humans, the number of glia constantly go up with evolution, with the maximum numbers seen in humans, and also depending on the region that you're talking about, the astrocytes or the glia in general outnumber the number of neurons present. So the number of glia we have is much more than the number of neurons we have. And among those glia, the major glial type are the astrocytes. Where are they produced? They are produced at the region where there is a lot of dividing neural stem cells. This is called the ventricular zone in the brain. These are ventricles like you have in the heart. There are ventricles in the brain as well. There are 4 ventricles. And surrounding these ventricles, you have the neural stem cells present, and these are the ones that divide. And in both in rodents and humans, the time course of cell development is the first cells to be born are the neurons, and once the neuron numbers build up, then the same stem cell switches fate to forming astrocytes. And once the astrocyte number reaches enough numbers, then the stem cell changes its fate and starts making oligodendrocytes. So it's the same mother stem cell that first makes neurons and then astrocytes and then oligodendrocytes. And once they are made in this narrow domain of ventricular subventricular zone, these cells migrate and then occupy different regions as they go along. And of course, over time, they also undergo more changes in their morphology and functions to suit which region they are occupying based on what they need to do locally. Like I said, these have a variety of functions that these astrocytes do. They provide nutrients to the brain. They maintain the blood-brain barrier. They regulate cerebral blood flow, pH homeostasis, regulate the extracellular ion concentration, and one of the more important functions is scavenging excess glutamate from the synaptic cleft. A major neurotransmitter in the nervous system is the amino acid glutamate. And the moment the glutamate is released into the synapse, once it activates the postsynaptic receptors, NMDA, etc., the glutamate has to be immediately removed from the synaptic cleft, because if the glutamate stays on for longer, it's going to have continuous effect on the postsynaptic neuron, and that's one of the reasons you have epileptic seizures, because of excess glutamate that is present, that is constantly activating the postsynaptic neuron. So the glutamate has to be sopped up, removed as fast as it possibly can, and that work is done by astrocytes. And the astrocytes, if they lose this capacity to remove the excess glutamate, it has very bad consequences on normal neuronal functions. They play a role in neuronal differentiation. The stem cells I spoke about a minute ago are depleted usually once all the oligodendrocytes are formed, and in the adult mouse, in the adult system, the astrocytes serve as the neural stem cells when there is a need for generating cells locally. They play a very important role in axon guidance, axon growth and guidance, synapse formation and elimination, and also in neuroinflammation, and this would be the major focus of my talk. Among all of these varied function, the one function that has really gathered attention in the last several years is this role in neuroinflammation. When the brain encounters a disease, when it encounters damage and infection, how do these astrocytes respond, and how do they function in either providing benefits or detrimental effects to the brain? And this physiological change in the status of these astrocytes is called the phenomenon called as reactive astrogliosis. So these are called the reactive astrocytes, and any physiological challenge to the cell converts them to a different stage. And in normal state, the cells have a particular morphology that we have seen, but when it undergoes this transition called as astrogliosis, and this can be in response to any sort of a tissue damage. If you have a stroke, if you have a concussion, sort of an injury to the brain, or if you have Alzheimer's, Parkinson's, Huntington, any sort of these multiple sclerosis, these astrocytes undergo a variety of changes, both at the level of gene expression, morphology, physiology, and they are much larger in size, and these are called the reactive astrocytes. And they also become impaired in some of the normal physiological functions that they do in terms of supporting the cells in the nervous system. And the reactive astrogliosis is disease specific. What you see following, say, a ischemic stroke, those reactive astrocytes are found to be very different if you have a traumatic brain injury, or in instances of Alzheimer's or Parkinson's or motor neuron disease. Even though they become reactive, they have completely different functions or changes that they undergo. And very widely thought idea is this: astrocytes becoming reactive is actually a maladaptive process. That they actually are not good in supporting the normal function, and so much so a lot of therapeutic approaches recently that companies are trying to spend on or look into is whether you can block these reactive astrocytes from forming, and whether that would be a beneficial mechanism to use. The reactive astrocytes undergo a spectrum of changes depending on the intensity of the injury that they have. So if the injury is a very mild one, or infection is very mild, lasting just for a few days, they undergo this change to become reactive. And once that injury is repaired, and when the infection is taken care of, the astrocytes go back to their normal morphology and functions. So we call that as mild to moderate astrogliosis, which is what you see here. When they become reactive, they become much bigger, more darker staining for certain markers, and then they actually go back to their normal state in A. But if the injury is very severe, you have a massive stroke, you have a spinal cord injury that paralyzes you, then these astrocytes start to proliferate as well, and they form what's called as a glial scar. It's very similar to the scar you would form on a skin when you have a wound that slowly heals up. So as this wound heals up, these astrocytes form a very thick band around it called the glial scar, and that persists for months to years depending on the extent of injury, or sometimes lifelong. And what is the function of these glial scar? The glial scar primarily is to make sure that anything that is damaging to the normal brain, the healthy part of the brain or the tissue, is actually prevented. And these could be infiltrating macrophages from the periphery, or T cells that come in, and the factors that they release, or it could be a viral or a bacterial pathogen. So the astrocytes ensure by forming these scars that these rogue elements and cells do not get into the healthy part of the brain. So that's the major functions of gliosis that we know of. And the spectrum, as we saw, can be induced by several changes. The astrocytes are very sensitive. So any perturbation to normal brain function is sensed by these guys along with the microglia. And as you can see, the trigger could come from any of those many sources in the brain and through many different molecules. One is, of course, damage to the CNS tissue that would come about by injury or by disease. And all those factors that are secreted can make these cells reactive. If the blood-brain barrier becomes weaker by other diseases, there are certain viruses like the Japanese encephalitis virus and West Nile virus that actually break the blood-brain barrier to get into the brain. And When that happens, some of these serum proteins get into the brain, and that's immediately sensed by these astrocytes, and they become reactive to prevent that. You can have other inflammation that happens, infiltrating leukocytes, macrophages from the periphery also secrete a whole lot of factors, the interferons and the interleukins that can actually activate these astrocytes to become reactive. So you can actually see how primed these cells are to sensing any perturbation, any slight change that the brain encounters from its normal healthy environment is immediately sensed by these cells, and they undergo changes to take care of those maladaptive changes. So normally, if you look at the effect of these reactive gliosis, it's mostly beneficial. They remove the excess glutamate that you see coming from cells that are dying, the neurons. They help in the blood-brain barrier repair. They restrict the spread of infection from the site of infection to the healthy part of the tissue. But also, unfortunately, if this gliosis persists for longer than necessary, then that itself is going to be detrimental to the brain function, because these astrocytes themselves can secrete cytokines and cause neuroinflammation. And lastly, inhibition of axon regeneration. A major idea in the field, if you have a spinal cord transaction, if you get a injury, damage to the spinal cord, you're not going to have any axon growing to repair, because the axons get stopped by the glial scar, and it's predominantly thought to be an inhibitory environment for axon growth. And so one of the drawbacks, even though the glial scar formation is to prevent further damage to the healthy tissue, it also has as a non-permissive barrier for the severed axons to grow back. And that's why when you have a central nervous system injury, like unfortunately falling off and tearing apart of your spinal cord, you're going to have paralysis depending on how bad your injury is for the rest of your life, because there's no chance that these axons are going to grow back. We got interested in astrocyte biology while we are studying a transcription factor called serum response factor. It binds as a dimer to very specific sequences on the DNA, promoters of genes, and it regulates transcription by associating with a whole lot of tissue-specific cofactors. SRF deletion, when you delete SRF in every cell in the body in mice, the mouse dies mid-gestation. The gestation for a mouse is around 20-21 days. The mice die by embryonic day 9, because SRF is very critical for heart formation, for mesoderm formation. So these mutant embryos do not form the circulatory system, and so they don't survive. And we got interested, work done by my graduate student at the time, Paul Liu, in WashU. So we wanted to look at what is the role of this transcription factor in brain development. So we used this Cree-Loxp system. It's a molecular, it's a genetic engineering approach, where if you want to delete a gene of interest, what you do is you use these sites, 34 base pair sequences called the Loxp sites, come from P1 bacteriophage, and you flank the region that you want to delete with these two triangles that you see, which are the Loxp sites. And that will be one mouse. In our case, we targeted exon 1 of SRF, and then you cross it with another mouse that expresses this enzyme called Cre recombinase, which would then cause recombination between these two Loxp sites, deleting the gene that is in between, the part of the genome that's in between. In our case, we used a promoter for a gene called nestin, which is expressed in all stem cells in the brain during development. So when you cross these two mice, you'll end up getting a mouse in which this protein is deleted only in neural stem cells. Okay, so this is to circumvent the embryonic lethality that we saw in these mice. So there will be no SRF deletion in any tissue except the neural stem cells. So when we did that, what we found was very surprising, that SRF deletion in neural stem cells, like I said, the stem cells first form neurons, then astrocytes and oligodendrocytes. We did not see any difference in the number of neurons that are born, but what we see is the number of astrocytes and the oligodendrocytes formed in the brain are about 50% compared to the control mice. And of course, these mice die very early, within 18 hours of being born. We don't know the reason why they do that. We do see cerebral hemorrhage in these mice, but we didn't characterize much about this. But we didn't know if this decrease is actually a reduction or a delay in differentiation. So then what we did is cross this mouse, I'm just going to skip a slide, cross this mouse to another mouse. So this SRF floxed mouse is going to be the same. This is going to be the determinant mouse in telling us where you want to delete SRF. And this work was carried on by a graduate student, Monica, who's graduated now. So now this enzyme, Cre recombinase, is going to be driven by a GFAP promoter, making deletion of SRF very restricted to only the astrocytes in the brain and no other cells. The neurons will not be touched, oligodendrocytes will not be affected, microglia will not be affected. Only the astrocytes will lose this protein very early, starting embryonic day 6.5. So when we did that, these mice live normally. They have no overt phenotype that we can see. But when we looked into the brain and asked what is happening to these astrocytes, whether the astrocyte numbers go down, astrocytes don't form, etc. But what we were very surprised to see is the astrocytes become reactive. They become much larger in size. This GFAP is a good marker for identifying these astrocytes, because after a particular age, 3 weeks of age, this gene gets shut off expression in the astrocytes, in the cortex and other parts of the brain. And in some regions, it expresses to a very, very low levels. Whereas in the mutant mice, these astrocytes very robustly express GFAP, and one indication of that is these cells are becoming reactive, because that's when these astrocytes continue to express GFAP. This is again to show if you take the cortex, there will be no GFAP expression in these astrocytes because they shut off this gene. But in our mutants, the astrocytes continue to express GFAP very, very strongly. And in the hippocampus, another region that's involved in spatial memory, the astrocytes express very basal amount of GFAP, but that expression really goes up in our mutants, indicating that these astrocytes are reactive. We have published this since. We have confirmed that these cells are reactive by using other protein markers for reactive state. And interestingly, when we delete SRF very early and the astrocytes are reactive, typically, like I said, the reactive phenotype is a very transient process. It comes on and then goes off and everything is normal. But since this is a genetic deletion and the astrocytes have lost a protein forever, what we found was the reactive astrocytes continue to be in a reactive state throughout the life of the animal. We've shown here until 12 months. We have looked data until 2 years of age in the mice, and the astrocytes are stuck in this reactive state throughout. To recap so far, SRF deletion in neural stem cells affects astrocyte differentiation. And if you delete SRF in astrocytes, it leads to a reactive phenotype. But of course, for a developmental biologist, the question is, if you delete something very early during development and you see a defect, the problem always is it because of something being affected during development process that's having an effect? Rather, if a protein is important for reactive astrocyte generation, then even if you knock it out once the system is completely developed, should also give you the same phenotype, and that's when you know that there is no developmental deficit that's causing the phenotype that you are seeing. So we used a third Cre mouse. In this case, it's the same GFAP Cre that we use because it comes on in, very early. So we used a third line. It's the Cre is the same, the promoter is the same, but the Cre enzyme is modified by tethering another protein domain from the estrogen receptor. So estrogen, as you know, the receptor is bound to the membrane. The estrogen comes and binds to a part of the estrogen receptor called the extracellular domain. And when estrogen binds to the extracellular domain, the protein complex goes into the cytoplasm, into the nucleus, and activates transcription. So the estrogen receptor itself can act as a transcription factor. So what people did NIFLY is they removed every part of the estrogen and only kept the ligand binding domain to which the estrogen binds. And they attached it to this Cre recombinase, and they mutated certain residues, protein residues, such that your endogenous tamoxifen would not bind to this receptor anymore. Instead, you have this molecule called tamoxifen that binds to the receptor. Tamoxifen is an analog of estrogen, and in certain classes of breast cancer, depending on what the genetics of breast cancer is, if it is an EGF receptor mutation that is causing increased estrogen response, then tamoxifen is used as a chemotherapeutic drug for breast cancer. So this tamoxifen binds only to this mutated estrogen receptor that we have attached to the enzyme. So basically in this mouse, The Cree will be made and expressed only in astrocytes, but it will not delete the gene until you inject the mouse with tamoxifen. So only when tamoxifen is added, it will now cause the Cree enzyme to go to the nucleus and cause recombination and deletion of the gene of interest to happen. So this gives us a very nifty handle on when we can delete SRF. And so what we did was we took the mouse and we took 2 month old mouse. So by 2 months, all brain development is complete for the major part. So which means that the astrocytes are completely developed, they have gone to where they should, form connections, doing their job. And now we hit them with either oil, which is a carrier, or tamoxifen, the drug, and started looking at the brain. And what we see is starting around between 1 and 2 months, the astrocytes become reactive. You don't see of the GFAP expression in the same mutant mice that is not being injected with tamoxifen but injected with oil. But when you give tamoxifen, these astrocytes now become reactive as indicated by the expression of this protein GFAP. So this told us this SRF is a very critical protein that actually regulates this transition of the astrocytes from a normal state to a reactive state. And also in this mouse, the astrocytes, I mean, we've confirmed this with other protein markers. And even in this mouse, the astrogliosis persists throughout life. They're all injected at 2 months. When we looked at every age until 24 months, so you can see here, you don't see astrocytes expressing GFAP in the mutants that they very strongly express GFAP in the hippocampus as well. So even in this mouse, when you give tamoxifen very early, after every development is complete, the astrocytes become reactive, they stay the same throughout the life of the animal. So one of the first things that we asked is, now the astrocytes are very abnormal in morphology and they're probably their physiological state. So we wanted to know, do they perform their functions normally? The first thing we looked at is the blood-brain barrier. So if the blood-brain barrier is intact or not can be judged by injecting the mouse with certain dyes, which would leak into the brain only if the blood-brain barrier is compromised. If the blood-brain barrier is intact, these dyes will not get into the brain. So what we did was we took this dextrans it's a 10 kilodalton protein, fluorescent. So what we do is we anesthetize the mouse, inject it into the heart, let this dye circulate in the system for about 10 minutes, 5 to 10 minutes, and then we sacrifice the animal, look at the brain, and look at the blood capillaries. All the staining you see are the blood vessels in the brain. When we look at, is there anything outside of these, we don't see any green color outside of the blood capillaries, indicating that the blood-brain barrier is not compromised. As an example, what we did was we stabbed the brain with a needle, which would break the blood-brain barrier, and now you can see the dye spilling out. We used another enzyme, horseradish peroxidase of 44 kilodalton. We get the same result where in the mutant mice and the control, we don't see any leakage of the dye, whereas if you stab the brain, you can start seeing leakage as a control experiment. This is at very old mice as well, 15 months of age. The blood capillaries do not seem to have been compromised for the blood-brain barrier because we do not see this enzyme leaking out into the brain in the intact brain. So that told us that the astrocytes are not probably compromised for at least one of the major functions, which is the blood-brain barrier integrity. Work over the last 5 to 10 years have shown that the astrocytes can be good or bad depending on what activates them. So they can be called as neurotoxic, where they actually can kill neurons and oligodendrocytes, or they can actually be neuroprotective. And the neurotoxic astrocytes have been characterized about 5 to 8 years ago in showing that they secrete factors that kills neurons, cannot support normal neuronal function, and can actually cause death of neurons after exotomy. So the question for us is, what is the nature of our astrocytes? Are they neuroprotective or are they neurotoxic? At least we want to make sure that they are not neurotoxic. So we've looked at entire brain. We don't see any loss of neurons, no structure that is being altered, no synapse, no axon projections that are affected, that gave us confidence that these cells are not like the characterized neurotoxic astrocytes, that they are not secreting anything to kill neurons. When we looked at synapses, and these are proteins made by astrocytes that promote synapse formation, we see expression of these mRNAs in these astrocytes not to change, indicating that the pro-synaptogenic factors are not affected in these mutant mice. We looked at the number of synapses, contact points between neurons, and younger and older mice, and we do not see any difference.
We lost him. Hello? We lost him.
Yes, sir. The voice is lost. Naren, sir? Naren, sir, can you hear me?
Am I back on?
Yes, sir.
Okay, I'm so sorry. Okay. So I'll go back. So we checked the gene expression for known
Sir,
markers
your
that people.
slide is not seen.
Oh, let me start again. Sorry. How about now? Can you see it now?
No, sir. Slides are not visible. Now it is started, sir.
Okay, okay. So I'll probably go back a couple of slides. So these mutant mice having these abnormal astrocytes do not have any memory deficits that we have probed for. Then we looked at known markers that can classify these astrocytes as being good or bad. And we ran through RNA expression analysis by quantitative PCR. And we find that the genes involved in neuroprotection, these are astrocytes that change when there is a stroke given. So when we looked at those genes, we found more of the neuroprotective marker genes upregulated in our astrocytes compared to what people have characterized as markers for the neurotoxic kind. We have done our own RNA sequencing where we isolated these astrocytes from the control or mutant mice and looked at their gene expression profile. Interestingly, what we find is genes involved in response to inflammation, to bacterial infection, to viral infection. These are the genes that are very highly upregulated in our mutant astrocytes compared to the control. Among the downregulated one, we see genes involved in astrocyte differentiation that are down, which is known because we have shown that SRF regulates astrocyte differentiation. So those are the ones that are down. So we validated these and shown that a lot of these interferon regulated genes are upregulated in our mutant astrocytes as much as genes involved in inflammatory response. So when you look at inflammatory response in the brain after any infection or injury, you can have either a pro-inflammatory response or an anti-inflammatory response. So these genes help either in regulating the inflammation in a good way or in a bad way. And surprisingly for us, the three major markers numbers of genes that are pro-inflammatory, the interleukin 6, TNF alpha, and inducible NOS, are all not changing in our astrocytes, whereas the others that are playing a role in sort of a positive regulation and inflammation are the ones that are going up. So all of this sort of gave us confidence that the kind of astrocytes that we have in these mutant mice are definitely not neurotoxic, because we don't see any cell death, no change in neuronal activity or function. So of course the big question is, are these astrocytes neuroprotective? So that's what we did. We looked at a whole bunch of experiments, a whole spectrum of injury or disease to the brain, and we looked at three paradigms of injury. One is a traumatic brain injury, neuronal excitotoxicity, and a stroke model, and in two different neurological disorders, Alzheimer's and Parkinson's. So we first looked at traumatic brain injury, and one way we could do this is a penetrating traumatic brain injury. There are different approaches that we can take. Just for ease and availability of technical help and knowledge, we went about with the penetrating injury done by my postdoc Surya. What we basically do is open up the skull, expose the brain, and just poke a needle and take it out. You don't have to worry so much. The brain doesn't have brain receptors, so the animal won't know any pain of the injection, except the surgery to open it up, and of course it's under anesthesia and also a lot of painkillers. But the brain per se cannot sense pain because it doesn't have pain receptors. So once we inject, just put the needle, take it out, and then see how the brain responds. Because when you have this penetrating injury, the brain actually seals in the injury from inside out. The damaged portion on the inside starts to heal first, and then it closes on the top. So this is how the preparation is. We open up, we know which region we are inserting the needle, and it's in the same position in all mice so that we are very close to where we are doing it. And then we look at the cavity of healing. Initially it forms a cavity and the cavity starts to close. So we are looking at the cavity. This is sideways, this is top down. As you can appreciate, the wound healing or closure is much faster in our mutant mice compared to the control mice. That's quantified here, indicating that our mutant mice with these reactive astrocytes can heal brain injury much faster than the control mice does. And the next approach we took is neuronal excitotoxicity. This is a very, very common means of cell death in the brain when you suffer from stroke, when you have epileptic seizures, etc. So that's because the firing neurons, or also the neurons that are dying because of both injury and stroke, actually release excess glutamate, and that will start to affect the healthy tissue neurons, and they will start to fire because of so much glutamate, and after a while they will not be able to keep up the firing rate, and they start to die. And how do we do this? And one way to do this is to inject toxins that actually mimic glutamate, and one such toxin is called canic acid. So we do the same experiment. We inject the canic into the ventricles of the brain, so that the major structure that is affected is called the hippocampus here, which is involved in spatial memory. We measure the seizure activity. So the moment you give kainate, the animal goes into full seizure, epileptic seizure lasting for about an hour or so, and then they come back to normal. So the seizure will stop because the brain immediately adapts within that time to remove all the excess glutamate, and everything becomes normal. But the damage is already set in that within a week, cells specifically in the hippocampus will start to die, and which is what is shown here. This is where the needle had gone in. You can see the damage done by the needle. This is the ventricle into which we would have injected this canic acid, and now you see in both sides stain for a marker that picks up dead cells, and you can see this green staining is all where the cells have died. So you can see very nice, this is in the control mouse. And when we did this with the control and the mutants and looked at the cell death, and what you can see on the left side is control, you can see massive cell death in the control mice, and you see almost no cell death, negligible cell death in the mutant mice. And we've used another marker, tunnel staining, which picks up broken down DNA that's caused by apoptosis. You can actually see all the red dots you see are the nuclei labeled for DNA that's broken. In a normal cell, the DNA ends are all kept intact, nothing is exposed, but when the cell undergoes apoptotic cell death, the DNA starts to break down, the ends are exposed, and that's what we usually label to pick You can see cell death in all the regions in the hippocampus. We hardly see anything. It's quantified here. So we see very nice neuroprotection in the conditions of canic acid-induced neurotoxicity, which is similar to what you would see in a stroke model. Then we actually did the stroke model, and one of the model that we went is called the photothrombotic stroke. We inject the mouse with a dye called rose Bengal, and this dye is normally is fine. It doesn't really do anything, but when you shine a particular wavelength of green light to the dye, and the dye then starts to undergo changes, becomes oxidized, releases a reactive oxygen species, which will cause blood platelets to accumulate and start a blood clot. So what we basically do is inject the dye, and the dye is there throughout the mouse body, but we open up the brain, and in a very, very narrow part, we use a laser. This is a cold laser. This is not a hot laser, so it's not going to heat up the tissue. A cold laser of this green light is then shown on this window that we make, very narrow, about 1 mm window, for about 15 minutes, and then suture up the mouse, and mouse is fine. But this causes focal stroke. As you can see here, this is the infarct region where all the neurons would have died, and you can also see activation of astrocytes around the infarct area. And if you were to do it and then look at every section, every clearing that you see here in white are the dead tissue in the brain. And this is the control, this is the mutant. You measure the area of these white patch, will tell you how big your stroke is. In the control, we see the stroke is much larger compared to the area occupied by stroke in the mutant mice, indicating that our mutant mice stroke. So in 3 different models of injury, we were able to find that our mutant mice with these abnormal reactive astrocytes are able to have better protection. And then moving on to more neurological disorders that are more relevant in these times. And first we looked at is Alzheimer's disease. We crossed our mutant mice with another mouse. It's a mouse that has mutations in certain genes identified in human patients, and one of the hallmarks of Alzheimer's is formation of these beta amyloid plaques in the brain. So what we did was we crossed our mice and stained for these plaques in the brain in control. This is about 9 to 12 months of age, these mice. You can see a lot of plaques in the neocortex and the hippocampus. But when you cross it with the tamoxifen inducible line, where you delete SRF only in the adult mice, we see a very significant reduction in the number of plaques as quantified here. When you take our straight line, the line that deletes very early in development, that also has gliosis, we virtually see no plaques left at all. The only difference between this line of mutants and the last set of mutants is basically how long these reactive astrocytes have been around. In the one where the reduction is only about 50%, the reduction is there for about 6 months, whereas in the other line of mice, the reactive astrocytes have been there for about 9 to 10 months of age. So, and therefore, longer these reactive astrocytes are present, more efficient they become in removing the amyloid plaques. This is a very, very tricky experiment. You have to wait for months to get these mice and then to inject them and wait. And so we don't have enough mice to run behavior and other experiments. But this is very encouraging that these astrocytes can actually reduce beta amyloid plaque load in the brains of these AD mice compared to the control. And lastly, we looked at Parkinson's. There are different ways you can model Parkinson's disease in mice. One is to inject this drug called the 6-hydroxydopamine. The other is the MPTP model. And the third is the alpha-synuclein model, which is a protein that gets mutated in human Parkinson's that can mimic Parkinson's in mouse. We are going one by one right now. We have finished our initial characterization with 6-hydroxydopamine. So this is a chemical that you can't inject into the mouse because it cannot cross the blood-brain barrier. So we have to directly inject it. The region in the brain that's affected in Parkinson's is a very small area called the substantia nigra pars compacta, SNPC, which sends its dopaminergic projections to the striatum, and the striatum regulates motor control. So with the SNPC neurons are the first to die in Parkinson's, in human Parkinson's, and therefore they do not make contacts with the striatum and you start having motor control problems. So what we did was we took the control and mutant mice, injected them with 6-hydroxydopamine, and then after a couple of weeks, we stained them for an enzyme called tyrosine hydroxylase, which stains these neurons in the substantia nigra, and counted the number of cells. So you can see actually, these are different regions that we look at every region so that we are not biased in our analysis. So we can see reduction of cells in the control, but no reduction in the knockouts. And of course, in another plane, you virtually see barely any cells left, whereas we see a lot more cells left in our mutant mice compared to the control. Same thing shown here, the fiber projection, because the cells die in the substantia nigra, so the fibers will also start to die. These are axon fibers that will also die. And you can see in the side that we are injecting in the striatum, we don't see much of the fibers coming in, whereas on the other side, the contralateral side, because the 6-hydroxydopamine is injected only on one side and not on both sides, so that we can use the other side as a control. Quantified here, we don't see any cell loss in our mutant mice in the substantia nigra in response to SNPC. We are now extending this to the other models of Parkinson's as well. To finally summarize, so we have chanced upon a transcriptional mechanism that is necessary to maintain astrocytes in a normal functioning state, and in the absence of SRF, these astrocytes become reactive, and the reactive state persists throughout the life of the animal. But interestingly, the SRF reactive astrocytes are not toxic. They do not affect any aspect that we know of normal brain functions, learning and memory, LTP, synaptic plasticity, you name it. We don't see any difference. In fact, it's far more protective under a very broad spectrum of insults that we can throw at the brain. And that work is being written up, and we have filed a provisional patent, so we could target SRF for therapeutics to convert the astrocytes into a neuroprotective phenotype. So we are now looking at more closer mechanisms, trying to identify specific markers for neuroprotective state. We are doing genome-wide screens using CRISPR-Cas9 technology to actually identify more genes that would be regulating this trace state transition from normal to a reactive state. And I would say that this is the first discovery of a very broadly neuroprotective reactive astrocytes so far, and nobody ever has identified or described an astrocyte that actually do a variety of functions, because astrocytes in AD and PD, Alzheimer's and Parkinson's, have been shown to be more detrimental than supportive to normal neuronal survival. With that, I would like to thank a whole lot of people without whom this work would not have been possible. Paul Liu, my first graduate student at WashU, who started this work, and later was continued by Monica, a graduate student here at IASC. She's since graduated and a postdoc at Icahn Medical School in New York. Surya, a postdoc, is still continuing and finishing the Parkinson's work. Srividya, a technician that helped years ago, and she's now a grad student at sunny Buffalo. Sumita, technician, and Priya, that are helping with a lot of these qPCRs and cloning. Students that worked on or working on other projects. Funding from intramural funding from IASC, the DST, also the DBT IASC Partnership Fund, and also CEFIPRA, and collaborators that we have, particularly James and Vijaya at JNC here in Bangalore, who have been very, very helpful with the electrophysiology experiments, and people that shared very valuable resources, mice and reagents that are very useful for this work. Thank you very much for this opportunity and your time, and I'll be happy to take questions.